Hello students, how are you all? Hope that you all are fine as well as healthy. We are learning biology. In biology, we are learning genetics and chapter number 5. The title of chapter number 5 is the principles of inheritance as well as variation. So in this chapter, in previous lectures, we have completed about the monohybrid cross. Okay, so you know that the Mendel did monohybrid cross, and with the help of monohybrid cross we are able to understand the inheritance of one gene. And you know that the father of genetics is Gregor Johann Mendel. Okay, and Mendel did different type of experiments on a one particular organism which is known as garden pea plant. The scientific name of garden pea plant is Pisum sativum. It is Pisum sativum. So you know that according to Mendel, we can say that according to Mendel, one character having the two different type of factors. Okay, one is dominant trait and another is the recessive trait. So Mendel did different type of experiments in the different type of 14 true breeders, means different type of 14 plants. Okay, and total 7 characters. Okay, total 7 characters were observed. So in last video lecture we have completed about the different type of examples. Okay, different type of examples with different type of methods of the monohybrid cross. So you know that the result of monohybrid cross is very much important. So two types of ratios are there. First one is genotypic ratio and second one is phenotypic ratio. So the genotypic ratio in monohybrid cross for F2 generation is 1:2:1, while phenotypic ratio of F2 generation will be 3:1. Okay, so before starting today's lecture, we will write the result of F2 generation of monohybrid cross. Okay, so we can say that here, if we write the result of monohybrid cross. Okay, result of monohybrid cross. You know that the monohybrid cross is done for the understanding of the inheritance of how many genes? Inheritance of one gene. Okay, inheritance of one gene. We can say that result for which generation? Result for the F2 generation. Result for which generation? Result for the F2 type of generation. Okay, so two different type of ratios are there. If we talk about the first one, which is known as genotypic ratio. If we talk about the first one, which is known as genotypic ratios, then we can say that for monohybrid cross for F2 generation, 1 capital T is there, is to 2 capital T small t is to 1 small t is equal to 25% capital T is to 50% capital T, small t is to 25% small t. It means 1/4 capital T is to 1/2 capital T, small t is to 1/4 small t. Okay, in short, you only remember the genotypic ratio is what? The genotypic ratio is 1:2:1. Okay, so that's all you will remember. Genotypic ratio is 1:2:1. Now if we talk about the second type, which is known as phenotypic ratio. So second type of ratio, which is known as phenotypic ratio. Second type of ratio, which is known as phenotypic ratios. Then what about the phenotypic ratio? You know that the from genotype phenotype will be decided. Okay, from genotype phenotype will be decided, because genotype means what? Genotypes means the gene. Okay, so gene will decide the character, or the gene will express, and then after expression of gene, character will be there. So if we talk about the phenotypic ratio, then 3 tall plants, okay, is to 1 dwarf plants. Okay, 3 tall is to 1 dwarfed. It means we can say that 75% tall plants is to 25% dwarf plants. It means we can say that 3/4 tall plants is to 1/4 dwarfed plants. Okay, it means in short you will remember the phenotypic ratio will be 3:1. Okay, phenotypic ratio will be what? The phenotypic ratio will be 3:1. So this both are the result of the F2 generation for the monohybrid cross. Okay, which is used for the understanding of the inheritance of one gene. Okay, so today in this video lecture we will talk about the new question, and the title of today's question is about the test cross. Okay, title of today's question is what? It is the test cross. Okay, see here, it is the test cross. Okay, see, it is the most IMP question of 2 mark. It is the question of how many marks? It is the question of 2 marks, but it is the most important. It is the most IMP question. 100% it will be asked in examination. Okay, first point, Mendel did, or we can say that test cross was done by Gregor Jonathan Mendel. Test cross was done by Gregor Jonathan Mendel. So first point, it is a one-liner question. The test cross was done by blank. We can say that test cross was done by Gregor Jonathan Mendel. Okay, point number 2, we can say that with the help of test cross, or why test cross is important. Okay, in point number 2 we are going to write the importance of test cross. We can say that with the help of test cross we are able to know about the unknown. We are able to know about what? We are able to know about the unknown genotype from given phenotype. Okay, so point number 2 is very much important. In point number 2 there is a importance of test cross. Okay, why test cross is important? So test cross is important because with the help of test cross, okay, with the help of test cross we are able to get what? We are able to get unknown genotype. Okay, if genotype is not known, means if genotype is unknown, then test cross is used. Okay, from what? From the given phenotype. Phenotype means what? Phenotype means character. Okay, see, tall is character. Okay, tall is phenotype, while capital T or capital T small t is genotype. Dwarf plant or the dwarf is phenotype, while small t will be what? The small t will be the genotype. So 2 points are completed about the test cross. First point, we can say that test cross was done by Mendel. Point number 2, what is the main function of test cross? With the help of test cross we are able to know the unknown genotype, okay, from the given phenotype. It means we can say that the phenotype is must. Okay, without phenotype test cross is not possible. So if phenotype is given, then yes, we are easily able to get the genotype. Okay, point number 3, we can say that Mendel did what? Point number 3, we will write this point about Mendel. Mendel did cross between tall plants of F2 generation with homozygous recessive. Okay, so if we talk about the height of stem, then homozygous recessive is equal to what? Homozygous recessive is equal to small t? Yes or no? Homozygous recessive is equal to what? Small t? See, it is a homozygous pair because the both alleles, okay, both alleles are same. If both alleles are same in a pair, it is known as homozygous pair. And small t is what? Small t is recessive. That's why it is known as homozygous recessive. Okay, see, so cross was done between what and what? Cross was done between tall plants of which generation? Of F2 generation. Okay, so you know that if we talk about the result of monohybrid cross, okay, result of monohybrid cross observed by Mendel observed that, see here, Mendel observed that in F1 generation 100% tall plants were there. Yes or no? Means capital T. While in F2 generation both tall and dwarfed plants were there. Okay, tall means capital T and capital T small t. Dwarfed means small t. So according to this result, tall means what? According to this result, tall means capital T or capital T small t. Okay, see, it is what? It is homozygous dominant. It is heterozygous. Okay, so see here what is given, we can say that tall is genotype or phenotype? Tall is not a genotype, it is phenotype. Tall is what? Tall is the phenotype. Okay, so we can say that from test cross, if phenotype is given, then we are able to know about the genotype. Okay, it means this plant in F2 generation may be capital T. Okay, it should be capital T or it should be capital T small t. So only two options are there for the tall plants. Only two options are there. Okay, either capital T or capital T small t. If capital T is there, it is known as what? It is known as homozygous dominant. It is known as homozygous dominant. What about this capital T small t? It is known as heterozygous. Okay, so Mendel did experiment or Mendel did cross between tall plants. Okay, see, still genotype is not known, only phenotype is there. Tall plant cross with the homozygous recessive, so that is fixed. Homozygous recessive, that is fixed. Okay, so Mendel said that, see here, Mendel said that if we get the result, we will remove this. Okay, so Mendel said that point number 4, if first option, if the result of test cross is 100% tall plants. See, we are writing what? We are writing tall plants, means we are writing the phenotype. Okay, we are writing what? We are writing the phenotype. If the result of test cross, means we are talking about the result between this both. We are talking about the result between this both. If the result of test cross is what? Is the 100% tall plants, then given unknown genotype is homozygous. Means capital T. Okay, you know that the two options are there, either capital T or capital T small t. So of result of this cross will be 100% tall plants, then the given unknown genotype will be homozygous. Second option, okay, we can say that if the result of if the result of test cross is 50% tall plants, 50% tall plants and 50% dwarf plants, and 50% dwarf plants. Okay, see, again we are writing only phenotype. Okay, we are focusing only on the phenotype. Okay, if this type of result is there, then given unknown genotype is, heterozygous. Okay, it is what? It will be the heterozygous. Heterozygous means capital T, small t. Okay, so two different type of options are there. If you will get this type of result, then option number 1 is there, and if you get that type of result, second type of result, then option number 2 is there. Okay, now we are doing the calculation. Okay, if the option number A is there, okay, option number A, the unknown plant is capital T for the first option, and for the second option it will be the capital T, small t. Remember, capital T means what? Means homozygous dominant, and capital T small t means what? It is the heterozygous. Okay, means what? Means the heterozygous, and cross with small t, cross with small t. See, this small t is fixed. Okay, because unknown plant must be crossed with homozygous recessive. Okay, so we can say that, see here, if we produce the gametes from here, then capital T will be produced from this parent, and that parent will produce the small t. So capital T combines with small t, and zygote will be formed, and result will be capital T, small t. So we can say that 100% tall plants are there. Yes or no? So see option number 1, if the result of test cross is 100% tall plants, then genotype will be the homozygous. Okay, see, if you are getting this type of result, then this plant must be capital T, or the genotype of this plant must be capital T. Okay, and now see, what about this? We can say that here, for this, gametes will be capital T and small t, because it is a heterozygous. Okay, and from here gamete will be the only small t. Okay, if you use the Punnett square for this, then capital T, small t. Okay, and here we can say that small t, capital T, small t, capital T, small t, and small t. So we can say that these plants are tall and these plants are dwarfed. Means see here, 50% tall plants are there and 50% dwarfed plants are there. So if this type of results are there, if you are getting this type of results, then the unknown genotype must be heterozygous. Again, see, these two sentences are very much important for the test cross. See again, if the result of test cross is 100% tall plants, then given genotype is homozygous. Okay, homozygous means both alleles are same. And if the result of test cross is 50-50, 50% tall and 50% dwarf, means 50% dominant and 50% recessive, then the given unknown genotype will be the heterozygous. Okay, see here, result is 100% tall, the unknown genotype will be capital T. Okay, result is 50% tall and 50% dwarf, then unknown genotype will be the heterozygous. So it is all about the test cross. Again, we are going to revise this test cross. Okay, so see, first point we can say that what about the test cross? It was done by the Mendel. Okay, it was done by the Mendel, and with the help of test cross we are able to know about what? We are able to know about the unknown genotype. Unknown genotype from what? From the given phenotype. Mendel did cross between tall plants of F2 generation. See here F2 generation, this word is important. Okay, if you are writing F1 generation, then here only one option is there, because in F1 generation all plants are there. F2 generation, okay, cross with the homozygous recessive means we can say that cross of unknown genotype, cross of unknown plant with homozygous recessive, okay. And see, two options are there. If 100% tall plants, then it will be the homozygous, capital T, and if 50% tall and 50% dwarf, then it will be the heterozygous, okay. So this question is completed of test cross, but in your textbook another example is given for the test cross. In your textbook another character, okay, another character is used, and this character is about the color of flower. So first part of this question is completed, and in second part we will write what? We will write we will understand the same, okay, same question of test cross, but with the help of another character which is known as the color of flower, okay. So if we take a one character, and this character is what? This character is the color of flower. You know that according to Mendel, one character having the two different type of factor. First one is dominant and second one is what? Second one is recessive. So if we write what? So if we write the dominant character, then we can say that the violet, okay, violet color is dominant, while if we write the recessive, then we can say that white color is recessive, okay. So the sign is important for dominant. The sign is what? For dominant, the sign is capital W, okay. For dominant means for violet, sign is capital W, while for the recessive the sign is small w, okay. So what is character? Character is the color of flower, okay. Violet color is dominant, sign will be capital W, and white color will be the recessive, sign will be the small w, okay. Sign will be the what? Sign will be the small w, okay. See, we are using what? We are using test cross, okay. Why test cross is used? Point number 2, we can say that with the help of test cross, see here, with the help of what? With the help of test cross, we can get unknown what? Unknown genotype, okay. Unknown genotype from what? Unknown genotype from given phenotype, okay. So given phenotype is must, okay. So now we are writing some options or some possibilities, okay. So pair number 1, if we write the capital W, so it is genotype, then the phenotype will be what? The phenotype will be the violet, okay. Now see, point number 2, if we write the capital W, small w, it will be also violet, okay. See, it is what? It is the homozygous, it is heterozygous, okay. And third one, if we write the small w, then this type of plant will be white plant, okay. It is also homozygous, but it is homozygous recessive, and that one is homozygous dominant, okay. So violet, the phenotype violet having the two different type of genotype, okay. The phenotype violet having two different type of genotype. First one is homozygous, second one is heterozygous, okay. Suppose we have a one plant or we have a one flower, and the color of flower is suppose violet. So we have a one flower of pea plant, and color of flower is what? Violet, okay. Violet is what? Violet is genotype or phenotype. We can say that violet is the one type of phenotype. Violet means what? Violet means one type of genotype, okay. Sorry, phenotype, but the genotype is still unknown, okay. So we don't have any idea about the genotype, okay. It should be, okay, see here, it should be option number 1, it should be capital W, means we can say that homozygous, Okay, it should be capital W. Capital W means homozygous, or second option we can say that capital W, small w, which is known as heterozygous. So two options are there. Okay, either homozygous or heterozygous. Okay, but in test cross we know the phenotype Violet, but we are not knowing the genotype. Okay, so the cross is must of unknown plant with what? With homozygous recessive. Okay, means small w. Here the cross is must with homozygous recessive, means small w. Okay, so first thing here, see here, if we take this as parent, then the gamete will be capital W, and here gamete will be the small w. Okay, if both gametes combine with each other and will form a 100% capital W, small w. Okay, means we can say that 100%. Okay, 100% violet flowers are there. If 100% violet flowers are there, then unknown genotype will be the homozygous. Okay, see here we are going to produce the gamete. The gamete will be small w. Here again gamete capital W and the small w. Okay, if we do the cross, okay, see the first one, we can say that capital W and small w. It is the first one. What about second one? We can say that small w and small w. Okay, so this both are what? This both are genotype. What about phenotype? This plant is violet, okay, and this plant having the white colored flower. So 50% white color and remaining 50% violet colored flower will be produced. Okay, so if 100% dominant character is there, then unknown will be the homozygous, means capital W, and if as result we can get 50% violet and 50% white, okay, white is, then we can say that the unknown genotype will be the heterozygous, mean capital W, small w. So it's all about the test cross. You will remember, with the help of test cross we are able to know about the unknown genotype. Okay, from what? From phenotype. See, phenotype is must. Okay, without phenotype genotype is not possible. Okay, so the phenotype is must. Okay, and remember, for doing test cross always we must done cross of unknown plant with homozygous recessive. Okay, so today's question is 100% important. Okay, in examination it will be surely asked. Okay, so I hope that you all are getting this, okay, very much easily. So today's lecture is 100% completed. Thank you very much.